The overall goal of the following experiment is to differentiate human pluripotent stem cells, or HPSCs, into neurons using a multi-titer plate format. This is achieved by harvesting and plating a single-cell suspension of HPSCs into V-bottom 96-well plates to form embryoid bodies, or EBs, overnight. Next, the EBs are transferred into U-shaped ultra-low attachment plates containing differentiation medium to induce neuroectoderm formation. Subsequently, the EBs are plated onto Matrigel-coated dishes to promote the outgrowth of neurons. Results can be obtained that show the presence of typical neuronal markers around day 8 by immunofluorescence staining.
Protocols for differentiating human pluripotent stem cells into neurons are often complicated and tedious. Somewhat accidentally, we came across a very simple procedure for making neurons from human pluripotent stem cells. From a technical point of view, the main advantage of this technique over existing methods is that it's easy to carry out, rapid and cost efficient at the same time. Presenting the procedure will be Dr. Miao Zhang from the lab.
Prepare EB formation medium and differentiation medium as outlined in the written protocol. Grow HPSCs under feeder-free conditions, such as in MEF-conditioned medium on Matrigel-coated plates or using chemically defined culture systems. Under subconfluent, actively growing conditions, using a stereomicroscope with a sterile plastic pipette tip, mechanically remove spontaneously differentiated colonies. Wash remaining undifferentiated colonies with PBS and use Accutase containing 1X-ROCK inhibitor to digest to single cells. Pellet the single cells by centrifugation at 200 G for 2 minutes and resuspend in a small volume of EB formation medium. Use a hemocytometer to determine the cell titer. Add additional cells to the remaining EB formation medium to arrive at a titer between 40,000 and 80,000 cells per milliliter. Using a multi-channel pipette, transfer 100 microliters per well to a 96 V-bottom plate, resulting in 4,000 to 8,000 cells per well. Spin the plate in a swing-out plate centrifuge at 400 G for 1 minute to collect all cells at the bottom of the wells. Transfer the cells to an incubator and allow EBs to form overnight. The next day, under the stereomicroscope, collect EBs and transfer in a small volume into a 3.5 centimeter bacterial dish containing 2 milliliters of differentiation medium. Wash the cells by gently rotating the dish. Add 100 microliters of differentiation medium to each well of an ultra-low attachment U-bottom 96-well plate. Under a stereomicroscope, transfer EBs in small volumes from the washing dish into the 96U wells. Incubate the EBs for 4 days to allow neuroectoderm formation. On the 4th day, wash the cells in differentiation medium and seed them one by one into the wells of a Matrigel-coated multi-titer plate, such as a 12-well plate. The cells should be equally distributed within the wells to allow undisturbed outgrowth of neurons. Allow the EBs to loosely attach to the Matrigel for approximately 10 minutes at room temperature, then carefully transfer the plate to the incubator. Neurons will grow out from the plated EBs in a radial manner within the next 4 days. Previously, we showed that, in line with many others, EBs from HPSCs could be generated simply by replating aggregates upon routine splitting of HPSCs into low attachment dishes. This usually results in a wide distribution of cell sizes. Another problem is that EBs maintained in suspension tend to aggregate with one another, leading to even higher size heterogeneity. To circumvent this, single HPSCs were plated in low attachment wells, which resulted in no EB formation and cell death. However, combining the cell survival molecule Y27632 with polyvinyl alcohol, which has been shown to enhance EB formation from HESCs, allowed EBs to form from single-cell HESC suspensions overnight. Conveniently, EB sizes in this multi-titer plate format could be tightly controlled by seeding different numbers of cells per well, in contrast to using conventional techniques. After transferring EBs to ultra-low attachment 96U plates, neuroectoderm formation was induced using a small molecule cocktail consisting of FGF, ERK, TGF-beta SMAD2, and BMP SMAD1 inhibitors, which then allowed the cells to be plated on Matrigel to give rise to neurons. Initiating EB formation from around 8,000 cells yielded the best neuronal differentiation capacity, which is characterized by pronounced neuronal outgrowths from the plated EBs, reduced necrotic EB centers after plating, and high expression of sensory and pan-neuronal markers such as BRN3A and beta-3 tubulin. The overall neuronal differentiation efficiencies appear to be comparable to the original procedure, which was applicable to both HESCs and HIPSCs. Thus far, 3 independent HPSC lines, HES, as well as HIPS cells have successfully been tested in the multi-well procedure, whereas cell line-dependent variability in differentiation efficiencies can generally not be ruled out. This figure shows dissociated single-day 4 neuroectodermal cells that were plated out as single cells and successfully differentiated at approximately 60% efficiency.
Handling EBs in the multi-titer plate format requires a bit of training in the beginning, but after some time you will see it's quite easy.